have a quick question. Would you guys feel like it was very rude if I stop my camera because I have to eat?
Oh, no, not at all. Go ahead.
I'm still going to be listening. I'm going to get headphones. I'm still going to listen to you, but I didn't want to be like munching on my food while you're talking. I think that was really rude.
No worries.
Okay, so I'm going to stop my video and still listen to you guys and everything. I just didn't want you guys staring at me.
No, that's.
I appreciate that. I teach college undergraduates on Zoom every week, so I'm used to staring at a lot of black screens.
I feel really bad, but I'm just like, my husband just made this huge dinner, and so I'm like,
no, enjoy your dinner.
Yes.
Okay, all right, thank you. And then when I'm done, I'll come back on.
Professor, we'll give it a few more minutes. Unfortunately, we did get a late reminder out this afternoon, so a few more minutes,
No problem.
minutes, and then we'll just go ahead and start.
Sounds good.
So what's Providence looking like for the summer and fall? Any plans to resume in person? I work off at MIT, and they just announced that they're hoping to get everyone back starting in September.
Yeah, so our plans are pretty similar. They're hoping to get fully in-person teaching up and running again in the fall, but currently the Department of Health wants us to plan as if classroom capacities were still limited due to social distancing, which I think is reasonable. So there's been a lot of shifting of classroom schedules, and we've added evening sections, and like the schedule's a mess, and I do not envy the people working on that. Thankfully, I teach small-ish classes, so I don't have any issues as far as classroom size. But yeah, the plan is to go fully in person in the fall with some social distancing. So we'll see what happens.
Wow. Yeah. MIT, I heard possible plans that they were hoping to vaccinate every single one of the staff members and their families. So until the vaccines become a little more readily available, I'm not sure if that'll happen by summer, but hopefully by the fall.
Yeah, that's the hope. I hope students can go home, get vaccinated, and then come back to campus. That would be ideal, but we'll see how it goes.
Okay, why don't we go ahead and get started? And I'm sure more people will be joining in.
Sure. Do you want me to just jump right into it? Do you have any announcements or anything to share before I get going?
Nope, not at this time.
Okay, cool. So hello, everyone. My name is Rachel. I got my PhD studying honeybees at Tufts University. So you may remember me from, I think that was last month, I talked about dirty water.
Yes.
Yeah, my concept of time is like non-existent right now. I just rely on my Outlook calendar to remind me of things. So last month, I talked to you about some of my PhD research on dirty water and minerals or micronutrients. Today, I will be talking to you about some of my PhD research on pollen foraging, focusing on macronutrients, and I'll talk more about what that means. I'm no longer at Tufts. I now have a job at Providence College. So I'm really excited to be there, and hopefully I'll have an apiary on campus at some point. But for now, everything's kind of virtual, and it's a little bit difficult to figure out where facilities will allow me to put stinging insects on campus. So TBD on that front. But if you have any questions and you don't have a chance to ask them after the presentation, I'll give you plenty of time to ask questions. But if there are more than I can answer, please feel free to reach out to me via email. I'm also pretty easy to find online and on social media. So my PhD research focused on nutrition, and kind of the big broad aspect of this is you can't really grow and survive and reproduce if you don't have the right nutrients. And so nutrition really shapes everything in our living world, whether you are a slime mold, which is this yellow stuff over here that eats bacteria, or you're a vegetarian rabbit eating leaves that maybe your owners have given to you. It's actually my brother's pet rabbit. So nutrition shapes everything, but it's actually really, really complicated. And I think anyone who's gone to the grocery store and say, just walk down the cereal aisle, you are inundated with choices, right? So we get to go to the grocery store to get our nutritional requirements, although it can be complicated. But organisms don't really have this option. They have to really interact with the environment to find the food that they need to get the nutrients that they need. And there are aspects of the environment that make these interactions even more complicated. So one aspect is abiotic factors such as temperature or precipitation. And I'm sure you've seen this with your honeybee hives. If it's too cold outside or if it's too rainy, they're not going to leave the hive to look for food. And so maybe they lose an opportunity to get the nutrients they need on that day. Temperature also affects digestion. So the warmer it is outside, the quicker digestion and metabolism works, the quicker you can absorb those really important nutrients. And one of the things I'm really interested in, or I was really interested in for my PhD and continue to be interested in, is the natural variation in the food that's available in the environment, but also the variation between nutritional content of food items. So this is my goddaughter here. She's eating a ham sandwich on white bread. And as adults, we all know you can't actually get all of the nutrients you need by only surviving on ham sandwiches on white bread. And even if this was the healthiest food, such as broccoli, eating one type of food, eating broccoli for your entire life is not really good for you. So I'm really interested in how organisms might balance the food that they're collecting in order to get the nutrients they need. And this is where honeybees come in. So I often get asked, why did I start studying honeybees? And I often tell people, the honeybees sort of chose me. They're just a really great system for these types of questions. And I'm sure I don't have to convince you all that honeybees are awesome, but I'm just going to point out some of the really cool ways in which you can study questions revolving nutrition and the differences between foods and behaviors and preferences in honeybees. One aspect also I forgot to mention, both with honeybees and my goddaughter, is there is a social aspect to nutrition as well. So in honeybee colonies, the foragers are the ones collecting the food, but they're not necessarily eating all of the food they bring back to the hive. And in the case of my goddaughter, she didn't make the sandwich for herself. A grown-up made it and gave it to her. And so she kind of has to rely on someone else to give her those nutrients she needs, just like in a honeybee colony. So as I mentioned, nutrition is really complicated, especially if you're an organism that has to interact with the ever-changing environment in order to get your food. So foraging theory is not quite as simple as hanging out at a deer crossing, but rather organisms really have to kind of do this balancing act and deal with all of these environmental factors that we aren't always thinking about. So as I said, honeybees, like you all know, they leave the hive to collect their food. They bring pollen back to the hive. They bring nectar. They bring water. And honeybees are a really great system for this. One, because there's that really interesting social aspect. This is one of the reasons that drew me to honeybees in the first place. The oldest adults are the ones bringing food back to the hive, but they're not necessarily the ones eating the food they bring back to the hive. So you can see here we have a nice army green pollen pellet on this bee as she makes her way back home. So we have this social aspect. We can also track the foragers and what they're collecting. So today I'll talk a lot about pollen and how we collected data on the pollen the bees collect, but you can collect data on water as well as nectar foraging pretty easily. Nutrition is also complicated when we think about organisms that live throughout the year, such as our honeybee colonies. So I'm sure you're all thinking about what is going to bloom first. Where will my girls forage first? Where can they find their food? Summer and fall, summer and spring, late spring and summer are usually rife with choices for bees, right? There's a lot of flowers blooming. Fall, their options tend to dwindle. And obviously by winter time, there's nothing blooming for bees. And that's when they have to rely on their honey. So as beekeepers, we have to think about this temporal or seasonal variation in nutrition throughout the year and how we might be able to provide our bees with supplemental either diets or with just lots of choices of flowers. So right now you might have some crocuses blooming in your yard. I took this picture the other day in my yard. My husband said, "There are little purple flowers in the yard blooming." And I said, "Crocus." And he said, "Oh, you went outside to look at them?" And I said, "No, I just know they're crocus." And I went outside to look at them myself, and they made me really happy to see these purple flowers sprawled across the ground. And we did have some honeybees foraging on the crocus. However, I didn't get pictures of them. So this is one of the earliest flowers that's going to provide our bees with the pollen and maybe nectar. I tend to see more pollen foragers on crocus. They might have nectar too, but it's better than no flowers, right? So right now, this is kind of all our bees have. A little later in the spring, we'll start to see clovers and dandelions. So a lot of weedy species emerge. And this is one of the reasons I like to talk about doing less yard work to help your bees. So if you mow your lawn less often, especially in early spring, when these are some of the few flowers that are blooming, you're going to do a lot for providing not only your bees with food, but also all the other pollinators that are flying around. And this time of year is especially important for queen bumblebees. They're just starting to emerge from underground. And if they don't have the right nutrients, they can't even begin to start their hives. So we often think about supplementing our bees early spring and late fall when the pollen resources and the nectar resources naturally are less diverse. Honeybees also deal with the spatial variation of nutrients or food resources, and that's just based on the way in which we have commercial pollination services here in the US. So for commercial pollination, our honeybees are kind of trucked around the US to these different monocultures, depending what's in season. And a colony in one of these monocultures really only has one option for food. And a good example for this monoculture is almonds. So if you're at my talk last month, you may remember this picture. So the hives here in the middle of the almond orchard don't really have any other food choice than pollen and nectar from almond flowers. And honeybees can fly pretty far from the hive. They've been known to forage within a 10-kilometer radius of their hive if they really have to, comfortably. I think it was Tom Seeley's work shows they stay somewhere between 4 and 6 kilometers from the hive. So they'll go further if they have to, but they don't necessarily like to. But these almond orchards can get to be up to the size of the state of Rhode Island. I'm in Rhode Island. It's a small state, but that's a giant almond orchard. So if you're a colony in the middle of this orchard, you really have no other options. And these bloom seasons are often long enough for at least one round of brood to be reared on only one type of floral resource or only one type of food. And so this can't really be that great for our bees nutritionally. So I've talked a lot about nectar and pollen. Today we're going to focus on pollen and what pollen nutrition is made up of and what happens if I actually kind of restrict what the bees are getting from their diet. So honeybees, as you all know, they collect pollen in their pollen baskets. They bring it back to the hive where they do store some of it. So they don't store pollen in near as much amounts as they store honey. This is why pollen dearths are actually quite dangerous for honeybee hives. they never have that much pollen in store. It tends to go straight to the developing brood because pollen is a source of protein, which is super important for honeybee development. So proteins are made up of amino acids, and there are 10 essential amino acids for honeybees. And you'll see these on my slides as EAAs, or essential amino acids. Now, de Groot in the 1950s determined which amino acids were essential for honeybees and in what amounts, and I'll show you that information shortly. But I just want to let you know what I mean by essential. So essential means, in this case, in the nutritional sense, honeybees can't make these amino acids themselves. They need to get them from food. So they need to get these amino acids from pollen in order to develop properly as larvae. So you don't have to remember this list of the 10 essential amino acids. I'll share it with you if you want. It's also in that de Groot paper, the 1953 paper. But these 10 essential amino acids are pretty much essential for most living organisms. So we know they're really important, again, for that survival, that growth, and that reproduction. So I'm going to be focusing on these essential amino acids tonight, but I can't talk about pollen without mentioning what else is in pollen. It's also a source of trace minerals. So I talked a little bit about that last month. And these are known as micronutrients, or things that bees need in smaller amounts. So sodium, potassium, calcium, and magnesium. And in my talk last month, I showed that calcium is probably really important as bees get ready for the winter time, both in pollen and in their water sources. Pollen's also a source of fatty acids, which, if you think about the types of fatty acids doctors tell us to eat, those omega-3s, those are fatty acids. They're very important nutritionally. And then lastly, pollen's also a source of microbes. And people tend to get, I don't want to say afraid, but nervous when they think about microbes. We often think about the bad things microbes can do for a good reason, right? There are a lot of things that our hives have to deal with that are microbial. So American foul brood, chalk brood, nosema, those are all the microbes that give microbes a bad name. But there are also some beneficial microbes that are important for the health of the honeybee gut and can actually functionally help honeybees produce some other nutrients that they might not be getting from their food. So these are all really important parts of honeybee nutrition. But again, I'll be really zoning in onto those amino acids and pollen as a protein source tonight. So I mentioned earlier that honeybees can't get all essential amino acids from crop monocultures. So this is that quantitative data from the de Groot paper. And these are our 10 essential amino acids on the bottom. These are their 3-letter abbreviations. And again, you don't have to really know which amino acid is which, but what I want you to pay attention to is here we have, this is the percent amino acid content required by honeybees for proper development. So de Groot did all these painstakingly intense titration studies where he fed developing bees at different levels of these amino acids and tracked development over time. So this is research they'll only let you do in the 50s because right now they just want you to publish. But it's really foundational, necessary research for this kind of, for beekeepers and honeybees. So here we have our 10 essential amino acids and the percent the honeybees need them in for proper development. If we look at what the percent amino acid content for almond pollen is, we can see there isn't any amino acid that almond pollen has more than honeybees need. They might get enough lysine from almond pollen, and it's completely missing tryptophan over here. So almond pollen alone isn't giving honeybees, or cannot give honeybees, the amino acids they need unless they forage like crazy in these monocultures. So they have to collect a lot more pollen to hit those necessary levels of amino acids for proper development. Another example of a common monoculture is sunflower pollen, which actually does a lot better than almond pollen. You can see it actually surpasses what honeybees need in some of these amino acids. It does have some of that tryptophan, which almond pollen was lacking, but it doesn't really get anywhere close to what honeybees need. And so these are just two common monocultures, but again, honeybees can't get everything they need from one or the other. So they really have to partake in this, we call it in ecology, we call it food switching. So they have to switch what they're eating in order to actually balance their diet. If we break down almond pollen and sunflower pollen into crude protein content now, so almond pollen, not looking at the specific amino acids, has 18% crude protein, and sunflower pollen has 13% crude protein. So here, when we look at these data, we're getting kind of different information. So almond pollen has more protein in it, but it doesn't necessarily have the right protein. And sunflower pollen has less protein, but it hits more of the marks that honeybees need. And so a lot of my PhD research was really focusing on protein quality rather than protein quantity. So it's not just about that crude protein content, but it's about what amino acids are actually in there making up the protein. So for the study I'll talk to you about today, I hypothesized that the lack of these dietary amino acids will affect honeybee foraging behavior as well as colony growth. So I will present kind of three sets of data tonight, one on the bees, one on the flowers or the forage the bees are bringing back to the hive, as well as their foraging behavior, and the last one on actually what's happening inside or within the colony as a whole when we manipulate the amino acids they have available to them. So that's kind of getting at that social aspect of nutrition. Can a hive itself actually develop and grow on a diet lacking amino acids? So for my first prediction, I predicted that honeybees raised on a diet lacking amino acids will invest more in pollen foraging. So they need to work harder to get the protein those young larvae need. My second prediction is that honeybees raised on a diet lacking amino acids will collect more diverse pollen. So we did some microscopy and some DNA sequencing to see what flowers the bees were actually foraging on and bringing pollen back to the hive from. And then finally, I predict that colonies raised on a diet lacking essential amino acids will expand at a much slower rate than colonies in my other diet treatments, which I will show you in a moment. So to do this research, I raised bees in the field at the gorgeous Tufts Vet School campus in Grafton, Massachusetts. So I had 9 hives, 3 on each diet treatment one year, and then the following year I had more grant money. So I had 5 hives on each diet treatment the following year. So you can see here are my hives after the fresh install along this tree line, and the facilities at the Tufts Vet School were fantastic to work with and kept a nice unmowed perimeter near my hives. I think that also partially had to do with them not wanting to go that close to the hives. It's a great place to keep bees. So we raised the bees in the field on one of 3 diet treatments. The first was a kind of representative monofloral dandelion diet. So this diet, I call them semi-synthetic diets because they're made with plant-derived amino acids, but you'll see the way I'm feeding them to the bees is a pretty synthetic way. Try to replicate nature as much as possible. So my first diet treatment is my monofloral dandelion diet. And this has 6 of the 10 essential amino acids that honeybees need that I know bees can get from dandelions. And again, this is based on research way back in the late 1940s, where they looked at what the amino acid content of different pollens are. I also raise bees on what I call my polyfloral wildflower diet. So presumably, if bees are kind of allowed to forage naturally, they're going to partake in food switching and get all of the amino acids they need. So I call this polyfloral because I think that you need to have a polyfloral diet to get all of these amino acids. And then when given the opportunity, bees will actually do it. So this is a semi-synthetic diet with all 10 of the essential amino acids honeybees need. Again, the diet was made with plant-derived amino acids. And then we kind of mixed it together with a recipe that we made in the lab. And then our last diet treatment is our control treatment, which was no diet manipulation and foraged freely. So the two experimental treatments, they had pollen traps on their hives. So any protein they were getting during the time of this experiment was from me and not from the pollen they were bringing back. Whereas the control colonies were just getting whatever protein the pollen was providing for them when they came back to the hive. So we installed the semi-synthetic diet in the hive as it would normally be stored. So here on the left, we have, some nice colorful looking pollen stored in the hive. And on the right, this is what our semi-synthetic diet looked like. So to make this diet, like I said, we got plant-derived amino acids in powdered form. We mix them together in those ratios, excuse me, in those ratios that the group says are important. We mixed in some honeybee healthy to get some of those trace minerals in there. And then we sprayed the frame with honey. One, because it helps the powder stay in the frame when we installed it in the hive. And two, it kind of gave bees, a scent to associate with food, right? As if you just give them a white powder, they might not realize this is a nutritional source. So we sprayed it with honey. So we sterilized these drawn comb frames, sprinkled an amount of the semi-synthetic diet in the hive, and we fed them more semi-synthetic diet as they ate it. And my PhD advisor was actually shocked that the bees actually consumed this diet. So I felt pretty proud of that. But they did consume it. And again, we sprayed honey to kind of make them realize it was food. We also installed these frames kind of close to the brood frames. Again, so they knew there's protein here, there's brood there. This is what I'm going to feed the brood. So our semi-synthetic diet looks a little different than the natural diet, but it is stored in the same way. And we allowed bees to forage and trapped pollen on return to the hive with the Sundance pollen traps. So before any experimental diets were installed in the hives, we actually turned these traps on for a set amount of time so that the bees would consume whatever pollen was already in there, right? Because we can't control what was already in the hive. So we gave all of our hives this kind of pollen trap on treatment, even the control hives. So they consumed all of the pollen that they had in the hive before the start of this experiment. And what's really great about these Sundance pollen traps, if you're studying pollen foraging, is they have this nice big collection drawer where you can get lots and lots of pollen. So we collected pollen foraging in a few different ways. Today, I'll be showing you data on weight of pollen collected. So we would go out to the field, empty these drawers into big gallon-sized Ziploc bags, and then actually weigh how much pollen each hive brought back. And I love these Sundance pollen traps because the drawer is nice and big, so we don't have to check it super often. Some of the smaller pollen traps, you do have to empty sometimes multiple times a day when the pollen's really coming in, because pollen is moist and it can get moldy, and you don't want mold in your hive. So the Sundance pollen traps are great if you want to trap a lot of pollen. So we collected pollen from all of our hives. We also sat on the side of the hives and counted rate of foraging. So who's leaving the hive, who's coming back with pollen. But today I'm just going to present pollen weight. So we had 2 years, again, with different sample sizes for our diet treatments. And for these data, the weight of bee collected pollen, I'm only showing you our experimental diet treatments because our control treatments didn't have a pollen trap, so we didn't have this data for them. So we have mean weight of bee pollen collected in our 2 diet treatments over 2 field seasons. And in 2016, there was significantly more pollen collected by the bees raised on the 6 out of 10 amino acid diet. So the bees that were lacking essential amino acids foraged for more pollen than those that were getting all 10 amino acids in their diet. So I want to note that in no case did a colony not bring pollen back to the hive. So they're still foraging for pollen, it's just a matter of how much they're foraging for. In 2017, we actually didn't end up seeing a difference. So in 2017, colonies brought similar amount of pollen back to the hive, no matter which diet treatment they were on. So this suggests that yes, amino acids could or probably do modulate honeybee foraging behavior, but there's a lot of other stuff in pollen that's also important for bees. So those fatty acids, those microbes, those trace minerals that they're not necessarily getting from our semi-synthetic diets. And so this suggests that there's something in pollen that's nutritionally important for bees, whether or not they have all 10 essential amino acids. And one of the reasons we think we had this difference between diet treatments, or the difference between effects of diet treatments between years, is in 2016, we had a pretty intense drought in Massachusetts that we did not experience in 2017. So here is the state of Massachusetts. This bee here is about where my hives are in Grafton, Massachusetts. And you can see it was abnormally dry in June 2016, and June 2017, there was no risk of drought. In July of 2016, we had moderate to severe drought where the bees were that we did not see in July of 2017. The only abnormally dry place is off the coast down there, nowhere near my bees. And so this suggests that when food is scarce, the difference between our diet treatments became even stronger. So possibly the stress between those 2 diet treatments may have been even more. Those bees in 2016 may have been even more stressed by the pollen foraging than those in 2017. So this happens in field biology. You get different years with different results. But like I said, no matter what the diet treatment was, they never stopped foraging for pollen. So pollen likely has really important stuff in it, more than protein. So honeybees raised on a diet lacking essential amino acids did invest more in pollen foraging, specifically when there was a drought. And this could be drought conditions in general cause the bees to work harder to find nutritional resources, because with a drought, we also have fewer flowers. We also use those pollen samples to identify the flowers the pollen was collected from. So pollen has this fatty acid layer that kind of protects the grains. That's actually quite hard to digest. So bees actually do a little bit of fermenting of the pollen in the hive that makes it easier for the young larvae to digest. It makes it with a little bit of spit and a little bit of nectar. And what we can do is we can actually collect the pollen and digest it using some pretty harsh acid in the lab to get rid of that fatty acid layer. And once we've done that, you can look at it under a microscope, and different floral species have different shapes and sizes and textures of pollen grains. So you can see here, there's 4 or 5 different types of pollen grains in this image. This image was 400 times magnification. And this was actually done by a high school student from Lexington High School. This paper we're actually working on publishing at some point in the fall, hopefully. But Adam is now a biology major at Johns Hopkins, and I could not be more proud of him. So he did all this really cool microscopy work to identify which flowers the bees were collecting their pollen from. So again, another great use of the Sundance pollen trap. You can look at these mixed pollen samples under the microscope and see where your bee is actually going. In addition to the microscopy, we did some genetic sequencing as well. So we could actually look at genetically which flowers are the bees bringing back to the hive. And this is a really cool kind of newish way of doing mixed samples of pollen. And the Best Bees Company in Boston, Massachusetts, they will do this method. I think it was like $150 for you to get this done. It might be a little bit more expensive now, but if you send Best Bees a sample of your honey and about $150 check, they will do this pollen sequencing on your honey, and they'll tell you which 3 species of flowers are most prominent in your honey. And the way they can do this is because honey just has pollen in it as happenstance. This is a little bit hard to kind of keep the nectar completely separate from the pollen. So you can do this genetic sequencing on your honey to see what species of flowers bees are bringing back to the hive. And then what we did to look at the data analysis is we combined all of the floral species into one general measure of diversity, which we call in science alpha diversity. So there's a few different measures of diversity. I'm looking at alpha diversity here. And the idea is that the higher the number is, the more diverse the source of pollen is. So again, we expected that bees being raised on that diet lacking essential amino acids or having that 6 out of 10 essential amino acids, we expected them to forage on more diverse pollen sources because they likely have to do more of that food switching, more of that balancing. So those will show up in the light blue. And then in that dark blue is our 10 out of 10 essential amino acids. And we expect them to be foraging from less diverse pollen sources just because they already get what they need from the semi-synthetic diet we're giving them. And you can see on the x-axis here are 6 hives from one area of our field site. So I had more hives than 6 in 2017, but for the amount of sampling and sequencing I was doing, I only had enough grant money to do 6 hives. So these are different colonies of bees, but in the same location. So the 2016 hive A is in the same location as 2017 hive A. So presumably they have access to the same resources as the colonies in the year before them. So when we look at the 2016 data, that light blue, those colonies lacking essential amino acids, they tend to go for more diverse pollen sources, but there's no real statistical significance here. And you can see there's a lot of variation within and between colonies. So hive F here had a huge range of diverse sources they were looking for, whereas hive C had kind of a smaller range as showed by the distance between the outliers. So we didn't have a difference in diversity of pollen collected in 2016 or in 2017. But one thing I want to note here in 2017 now is we can see there's a lot less variation or distance between that bottom and that top point than we have in 2016. And so again, this suggests that our bees in 2016 were a little bit more stressed than our bees in 2017. So maybe in 2017, there are some really good pollen sources out there, and they didn't need to diversify as much as they did in 2016. And I still have these pollen samples in the freezer at Tufts, taking up a ton of space. And I will eventually move them to my lab at Providence College, where we'll do some amino acid analysis of these different samples to see nutritionally what were the bees actually bringing back to their hive. So there are a couple studies that show when bees are lacking certain amino acids inside the hive, they'll forage for them outside the hive. So even though the diversity of pollen collected doesn't differ here, maybe the nutritional content of the pollen collected differs. And I want to talk about the top 5 species that each diet treatment collected. So our 10 out of 10 diet treatment, the one with that polyfloral wildflower diet, they collected pollen from purple loosestrife, water lily, American plantain, red clover, and white clover. So we have our weedy species in there. And the bees raised on the 6 out of 10 amino acids, foraged on smooth winterberry, purple loosestrife again, ribwort plantain, black elderberry, and white clover. So these are the genetic sequences that showed up the most often in each diet treatment. So if we look at the similarities here, both diet treatments had a lot of purple loosestrife pollen and a lot of white clover pollen. And when I say a lot, I'm talking about relative abundance. So we're talking about proportions as a way to kind of control for our hives bringing different amounts of pollen back to the hive. So this could suggest that purple loosestrife and white clover are nutritionally important sources for bees. Purple loosestrife is not necessarily native, so I don't really suggest planting it. It does kind of just bloom naturally in a lot of places along the coast. But white clover, again, you can help your bees by just doing less yard work and letting that white clover bloom. I also want to highlight the differences between our two diet treatments. So the 10 out of 10s foraged on water lily, American plantain, and red clover. And the 6 out of 10s foraged on smooth winterberry, ribwort plantain, which is in the same genus as American plantain, but a separate species, and black elderberry. So it could be that these pollen sources have different amino acids than each other. One thing that actually really surprised us was the amount of water lily our bees were getting pollen from. And we thought that in 2016, our bees were getting a ton of water lily pollen because there was a drought and they were just going to nearby ponds to find flowers because that's all they had available to them. But these results were repeated in 2017. So water lily might be a good nutritional resource for bees. Not sure yet. One thing though, I do want to mention that's actually quite neat about the best bees, the honey sequencing, honey DNA, I think they call it. So all of the honey DNA samples they keep that sequencing data within a big database. So they're building a database of bees in the city and what flowers they're bringing back to the hive. And it turns out that bees that are raised or kept in Jamaica Plain have much more diverse pollen diets than bees that are kept anywhere else in the Boston area so far. And the reason is because the bees kept in Jamaica Plain are next to the Arnold's Arboretum, and they're bringing back some like crazy ornamental pollen sources that bees, say in Medford or Somerville, don't necessarily have access to. And so being in the city isn't necessarily bad for bees. I think sometimes we think about country bees maybe having more space and forage than city bees, but it could be that city bees might have more diverse forage than what we call our country bees, and it might be beneficial to them. So I want to dig more with my lab at Providence College into these differences between the nutritional content of both these specific floral species, as well as what the bees are bringing back to the hive. So honeybees raised on a diet lacking amino acids didn't really collect more diverse pollen than those raised on a diet with all 10 amino acids. And that could be for a number of reasons. Like I said, we didn't dig into the nutritional content. Maybe they're foraging for different nutrients specifically, or maybe they're looking for beyond protein, they're looking for those fatty acids, those micronutrients, those microbes even. And then finally, for this third set of data, we tracked colony growth. And we tracked a bunch of different factors of colony growth. Today, I'm going to show you just the number of frames fully covered in adult bees. And as you know, the more frames fully covered in adult bees, the more bees you have, the bigger workforce you have, both for foraging as well as rearing brood. So we tracked colony growth throughout the year in our hives. And we looked at the number of frames covered in adult bees. So here on our y-axis, we have that number of frames. And the x-axis here now, we have weeks since installation. So these were nucleus colonies installed in early May. So they all began with about 5 frames of adult bees, 3 frames of brood, and 2 frames of mixed food. And we controlled things as best as we could. It's not always easy to control the amount of bees you have. but these all began as nucleus colonies in May, were requeened on the Grafton campus. And then again, we counted the number of frames covered by bees. So you'll see our control treatment now, that treatment that didn't have the pollen trapped, that'll show up in gray. Our 10 out of 10 treatment with all the EAAs will show up in that dark blue still, and our diet treatment lacking essential amino acids will now be in that light blue. And if we look at our points, there's, again, you can see a lot of variation, especially in those experimental colonies, but we can use some fancy statistics to fit kind of a growth or a linear slope to these points. And so our dark blue treatment is about 1/3 the growth rate of our control treatment. So the bees in the controls treatment had a slope of about 1/2 frame a week. And the bees in our 10 out of 10 diet treatment, or our polyfloral semi-synthetic diet, had about 0.19. So the bees being raised in our semi-synthetic diet treatment are growing at about 1/3 of the rate of our control colonies. So again, pollen has other stuff in it that's important for growth in addition to protein. And the colonies that were raised on a diet lacking essential amino acids had a negative 0.01 slope, so pretty much 0, which means they weren't able to grow at all. So colonies without any or without all of the amino acids did not expand beyond those 5 frames from the nucleus colony. But again, we do have a lot of variation, especially in those experimental treatments. So colonies raised on a diet lacking essential amino acids did expand at a slower rate. And I think this is the strongest conclusion we have over these both years of data. Honeybees need natural forage to develop properly, and they need, at the very least, all 10 essential amino acids to grow at all as a colony. So colonies lacking the essential amino acids, why aren't they growing? A few things could be happening. One, the bees could be attempting to raise a new queen. So maybe this lack of development in the colony is detected by the workers as our queen's doing a bad job, we need to raise a new one. So we did look at the number of queen cells, and there actually wasn't a difference between queen cells in the 6 out of 10 hives and the 10 out of 10 hives. So we don't really have evidence that suggests they're attempting to raise a new queen, but that could be an explanation to look into. The workers in those colonies lacking amino acids could be partaking in cannibalism of larvae as a method of compensatory feeding. So when a hive is super, super desperate and they need protein from somewhere and they're not getting it from their flowers, they will cannibalize young larvae, feed them to older larvae as a way of kind of compensating for that lack of protein. And there may have been a high rate of larval death due to the lack of essential amino acids. We do have some data on the brood pattern and the brood area, so that's something that we could dig into a little bit more. For both experimental treatments, though, the general availability of nutrients may have been affected by those semi-synthetic diets. So there's other stuff in pollen that's probably also important. Like I said, amino acids, trace minerals, fatty acids, and even microbes. So what does this mean for our bees? The biggest thing I tell people all the time, beekeeper or not, if you want to help the bees, just plant diverse flowers that bloom at diverse times throughout the year. Because I believe that if honeybees are given the choices, they're probably smart enough to find what they need. So it's all about diversity. And even in the city, you can provide bees with diverse forage choices, even without a lot of space. So if you plant a few different flowers that bloom at different times of the year, you're giving the bees diverse choices to choose from. So some of my favorite resources for picking which flowers to plant. Again, you want those diverse pollen sources, you want diverse bloom times. And one of my favorite resources for looking into this is this book called Garden Plants for Honeybees by Peter Lintner. He worked at a botanical garden and did all kinds of observational studies and research looking at what the bees visited at what different times of year and what they tended to be collecting from these floral sources. So there's ratings for pollen and nectar, and there's bloom times. And so this is a really great book to think about planting nutritionally for your bees throughout the year. One caveat I'd like to say, this book, Peter Lintner did his work in a botanical garden. So there are things that don't necessarily or aren't necessarily native to your area. So you want to avoid that. But you also want to avoid these crazy cultivars. So believe it or not, sunflowers are a fantastic resource for all bees. They're just loaded with pollen and nectar. But there exists a cultivar of sunflowers that does not have any pollen in it because people don't like to have allergies to their flowers. And while this might be beneficial to us, as you might imagine, pollenless sunflowers aren't very good nutritionally for our bees. So avoid those crazy cultivars so that bees can actually collect pollen and bring it back to the hive. And I mentioned in the previous slide about native flowers. The Pollinator Partnership has some really great eco-regional planting guides that tells you what to plant and when it blooms based on wherever you are in the country. And you can download these guides for free. So I highly recommend those. And then the Pollinator Partnership in late June is doing this really awesome pollinator week to kind of bolster awareness for pollinator health in general. So you can partake in Pollinator Week as a bee club. You can just go online and register whatever events you're doing, and you'll get recognition for that. Or as an individual, you can register any kind of private events you're doing, and you get recognition, but it doesn't necessarily go on their Pollinator Week map. So this is something I'm looking forward to, and I always look forward to Pollinator Week. So with that, I'd like to thank everyone who helped me collect the data on this research. As you all know, beekeeping is a labor of love, and it's made a lot easier when I have a lot of interns that are able to help out. Joanna Chang and James Gonzalez specifically helped a lot with the beekeeping and the collection for this data. And then my funding sources, and while I was at Tufts, I got all of my bees and beekeeping supplies from New England Bees. I'm a little bit further away now, so I'm gonna have to switch my bee company, but I loved working with them, and I want to thank them and all my funding sources. And thank you all for coming, zooming in tonight, quote unquote, coming to my talk and listening to my research. I'd be happy to take any questions you have. And again, if I don't get to your question tonight, feel free to send me an email. If you want to ask, I think I saw some stuff pop up in the chat. I can look at the chat, or if you want to unmute yourself, I'm happy to.
Professor, I had a question about one of your slides on the percentage of protein in the pollen. I think it was one of the earlier slides. It was around 17% or...
the crude protein amount?
Yes.
Yes, go ahead, sorry.
And actually, would you mind pulling your...
Not at all. I know which slide you're talking about. Where'd my Zoom go? This one, yes?
I think it was one or two slides ahead of this one. Okay, so almond pollen is about 18% crude protein, and then water content. I assume carbohydrate or just plant matter.
Yep.
And okay.
Yeah, so fatty acids, carbohydrates, plant matter, trace minerals. Pollen is actually super complicated chemically, and that's why there's way more research on nectar than there is on pollen, just because the chemistry of it is way more complicated.
Okay, thank you.
Mm-hmm.
Hey, Rachel, I have a question.
Yes.
about the differences in the two hives, the 1010 and the 610.
Mhm.
And how, I guess, do you know, all other things being equal, like how do we know that, one of the 610 colony didn't have, fewer bees or a weaker queen, or we're battling something or other? I mean, it's very hard, I would imagine, to make sure all the variables are kind of kept equal when you're trying to do these type of experiments.
Yeah, I mean, that's one of the things I love about fieldwork is you have to really pay attention to the details, but also sometimes get creative. When we were checking on the hives, we checked on them twice. We checked on them weekly. We opened them up, and then twice a week we looked at foraging behavior. And we really paid attention to any signs of disease or if the queen was laying. But like I said, in those 6 out of 10 hives, they weren't growing. And so they definitely didn't have as much brood as the 10 out of 10 hives. And so it could be that by some chance, all 3 of those hives just had a weak queen. But it's really hard to say for certain, other than we got our queens from the same supplier. Everything was installed at the same time, and we really paid attention to see if there were any signs of disease. So we didn't have any signs of disease throughout this experiment. We treated for mites after and before the experiment. So we kind of had that baseline everywhere, but we didn't do any treatments during the experiment. We just paid attention to what was happening in the hive. So yeah, it is always hard to really control things outside, but I find the results to be a bit more meaningful, right, than raising things in the lab, as necessary as those are to kind of build up to the field experiments. I have a lot more fun outside.
Thank you.
Amanda had a question for you, Professor. From what I gather, which in addition to diverse flowers for bees, should I supplement my supplement feeding with honeybee healthy, or I assume artificial protein patties?
Yeah, so this is a struggle I have with. I think supplemental feeding is often important, especially if you're in a dearth or if it's wintertime and your bees don't have what they need. The only thing that I'm kind of wary of when it comes to anything that has kind of a supplemental amino acids is if you look at the back of most protein supplements, they just have all 20 amino acids in them. And we don't necessarily know if bees need to be fed all of those 20 amino acids. We don't know if they can digest all 20. Maybe we're actually giving them more work to process and excrete some of those amino acids. And so while I do think supplemental feeding is important and you do want to be on top of it if your bees don't have any pollen, keeping their diet as natural as possible throughout the year, I think, is the best way to go. I do hope to do some experiments on those diet supplements once I get an apiary up and running at Providence College to see just growth on those diet supplements. I haven't told my undergrad yet, but one thing I want her to do, we can, put a white kind of sheet in front of the hives and we can measure how often they are excreting waste. So I'm not certain that those supplements necessarily have everything the bees need, but they're not going to hurt them necessarily. So I'm always saying keep your bees' diet as natural as possible, but I also recognize there are times when you just have to supplement. I used Honeybee Healthy in these diets specifically because they have those trace minerals in it that the bees might need, and they're not necessarily getting it from everywhere else. I think if the bees are given choices, they'll find what they need and they'll figure it out. But I know that's easier said than done, especially in New England.
Excellent, thank you. Joanna had the next question about whether pollen from vegetable plants provide different essential amino acids. They have a different essential amino acid profile than non-edible flower plants, and is one better than the other in terms of location? I assume locating or hive placement.
Yeah, that's a fantastic question, and I'm not really sure the answer to that. I suspect the profile is similar in that it's not going to have everything the bees need. It might have more than almond or sunflower pollen, but I'm not certain. Like I said, there's really very little done on pollen nutrition. So it'd be interesting to look at, even in community gardens, what's in the vegetables, the crops that give up vegetables and fruits versus those that are sunflowers and cut flowers, etc. As far as positioning your hives, anywhere there are flowers, I think will do them well. I say anywhere there are flowers, but there are a couple flowers that are actually toxic to bees because of the sugar they create. And I can't remember off of the top of my head which those are. I think one might be oleander.
Rhododendrons?
Rhododendrons, definitely. Yes. There's another one. But again, if the bees are given choices, they're not going to go to the rhododendrons if they have better options nearby. If that's all they have, then they're probably going to end up there, unfortunately. So giving them choices is always good. I always tell people, you know, vegetable flowers are great because not only are the bees getting something out of it, you're getting something out of it too, right? More bees in your garden are only going to give you more fruits and vegetables. But yeah, I'm not certain if the profile is similar or different. It'd be an interesting chemical question to look into.
Excellent. If anyone has any more questions, you can go ahead and unmute yourself. Marty?
Yeah, thanks. I was interested in the purple loosestrife that you all talked about, and I seem to recall that there was some kind of insect that they added to control purple loosestrife in watered areas to actually reduce the purple loosestrife from growing in a given year, a given time period.
Super interesting.
And apparently it has been very successful.
Good to know. Yeah, purple loosestrife is invasive, so you don't want to plant it.
Also, there's a person in Maine who has some 5,000 acres where he is planting purple loosestrife because he likes the honey, and he was given permission because he has enough land in order to do that.
Yep. Yeah, I mean, if you can control it, it's not always easy to control a plant. But I wouldn't suggest planting it in any urban garden or backyard garden. It's a great nectar source in the fall. Surprising. So it's not all bad, but we don't want to promote invasive things. And
one more thing.
Yeah.
Ken Warchall, our beekeeper from Worcester County Beekeepers, he takes pollen and puts them into frames and gives it to his bees in the spring.
Oh, look at that.
As a bee supplement that he put together.
Yep. So more natural, not necessarily throwing everything that the bees might possibly need at them, but rather what the bees actually collect. That's a great idea.
Thank you.
One thing to be careful of with feeding hives pollen is like transfer of microbes. But I suspect Ken knows what he's doing because he's been at it for a long time. So just if you're refeeding pollen between colonies, just be aware that microbes can be passed, whether they're good or bad.
So, anyone else?
Sorry, does that imply that the microbes are in the pollen or in the pollen bee bread that's between the frames that you were thinking of?
So the microbes can be in both. There are studies of actually honeybee diseases getting passed to bumblebees from pollen in the field. And so we know microbes can kind of survive on the pollen. But if you freeze the pollen, you're probably killing a lot of the stuff that's living on the pollen. So that's one way to kind of sterilize the pollen before you refeed it to another hive. But there are more microbes in bee bread. That is true because it's fermented pollen.
Excellent.
One more thing. Sorry.
No problem.
There was on a different bee meeting, they had a person with an electron microscope.
Yep.
And he was looking at bees but he was also looking at some of the pollen bread that was in there. It was very fascinating how it looked like coffee beans to me,
Yeah.
to some of them.
Yep.
But it was very interesting. Thank you for your time.
So,
Thank
Professor, just following up on that, you said you have some older samples in the deep freezer off at Tufts, and you're hoping to identify some of the pollen sources from your 2016, 2017 studies. How will you go ahead and identify those?
So I'm really interested. So we did some genetics and some microscopy to identify the flowers, but I'm also interested in getting some nutritional kind of levels of the pollen. There are a few different ways to get amino acids from the pollen. And so far, what I found, they all involve liquefying the pollen, which is more difficult than working with pollen as a solid. So I have to liquefy the pollen, and then you can do some gas chromatography, some HPLC, which is like stuff I did way back in Gen Chem as an undergrad. It's basically you use flames and things are different colors, or they move at different speeds along a special piece of paper, depending on what protein they are. So that would be amino acid content. I'm also just kind of interested in that crude protein content because it is a lot easier to get. So you can get carbon to nitrogen ratios pretty easily with this really cool machine that combusts the pollen. So again, some crazy chemical reactions are happening to get those nutrient contents. So we'll see if I have some more data to share maybe next spring on some of those samples.
Excellent, fascinating. Well, thank you everyone for attending. And two more messages in the chat. How do you know which sunflower is good for bees, as opposed to, you mentioned that there was one cultivar that is not?
Yeah, so the cultivar that's pollenless, I cannot remember the name. I should look it up. But it usually, if you look in any of the catalogs, like I get the burpee catalogs all the time and I look through them because I like flowers. They'll say in the description if it's a cultivar, what kind of cultivar it is, and if it's pollenless. So the pollenless cultivars are usually pretty heavily advertised because people want them for lack of allergies. So just make sure you read those descriptions before you buy seeds or plants. Also, if you're at a garden center, you can, on a good sunflower, you can literally see like the yellow pollen on the anthers. So if you're at a garden center, you see pollen on a plant and bees are visiting a plant, it's probably good for bees. So that's one way I like to pick my flowers. I just kind of walk around the garden center and see where the bees are going. And then I read the description, see if it's native, and all of those things. So if you just walk around, check it out.
Excellent. And one final question. Joanna asked, was it Boston Best Bees that does the pollen sequencing?
Yep, Best Bees Company.
Best Bees Company.
So they're based in Boston, but they do have some locations throughout the US, and you can mail your honey to them. So it's Best Bees Company, and it's called Honey DNA.
Excellent. Well, thank you everyone for joining us, and thank you, Professor. We
thank you so much for having me.
Updates on your research sometime in the future.
Yes, I hope I have an apiary soon.
Excellent. Have a good evening, everyone.
You as well. Thank you.